do it that way. So this is a good way that you should possibly be utilizing this particular feature. Okay, so yes, you should probably move through them sequentially one at a time, but if you get to the point where you need to go back and check some questions that you've jotted down in your notebook, this is a much quicker way you can do so, rather than have to go through 70 of them. Okay, now remember, I'm not sure, no one really reported this as an issue on the Facebook page for 2803, but I just spoke to Karina, who's the head of program, and she did say that a couple of students got to the last question and for some reason it saved and submitted them out because there was a forward arrow. But as you can see here, there's no forward arrow. You can only go back. So the only way you can really leave the exam at this point is to click that. So let's just say you got to your 80th question, not 20th, 80th question, and you still had 10 minutes to go. You still have the potential to go back to any question you want to, but if you select that, as soon as you put that, it's finished. Okay, so please ensure you don't press that until you're really ready to go. Okay, and you can decide if you want to go back one at a time, like this, or you want to jump through right to the start again, like that, or you want to select the one most specific to you. Okay, so I'll close that one out. Hopefully that makes sense. So I definitely would be utilizing that option for Monday. Okay, so let me leave. Okay, so guys, do you have any questions in terms of just the exam logistics, not what's in it, just anything around that side of things? Unfortunately, I can't get my camera to work, so you can't see me, but you've got the screen there. Anyone want to pop any questions up into the chat? I ask a question, how to relieve adverse. We'll get into specifics in a second, but does anyone, in terms of content, does anyone have any questions about the logistics of how the exam will run? So basically when you guys log in at 7:45, you'll see this. If you haven't yet, please watch this video, very important. If you haven't already, please attempt the practice exam. Okay, so you'd be crazy if you haven't attempted the practice exam by Monday. You'd be crazy if you haven't watched the video and just have a read through this information. This will give you information about technical advice. So as I've said in my previous captures, please make sure you have a reliable internet connection. If you don't, you can please try to access one, or you can even go to the university and use its Wi-Fi. It's very important that you have good, reliable, relatively fast internet. If you don't, it's questionable. Please try to look into that ASAP. Please also ensure that you use the right browser. Don't use Internet Explorer, preferably Firefox or Google Chrome. Make sure you clear your cache. Okay, I sent you an email with that information ahead of time. And then on the day, okay, so this is the options that you do have on the day if something goes wrong. You can, considering that all you guys are probably Gold Coast students, you can contact my office, so call my office. So please, ahead of time, have my number written down, my office number, just so you know it's 555 28 759, but have that next to you. So call that if you have any issues with the IT side of things. If that doesn't work, just screen capture what you're currently at and then email it to me and just say, hey, Matt, I'm currently having some issues. Can you please have a look into this? Try to rectify it yourself, but if you can't do it, I'll try to come in and assist. And then if we're really having difficulties, we'll bring in IT. But just that's the third port of call. Now, saying that, for 2803, now this was sent to me by the head of program, 96.8% of students finished the exam. So that's telling you that really next to no people had significant issues from an IT perspective. I know a number of you have reported that you had issues with lagging and so forth, but this may come down to the speed of internet. So maybe just that's something, and the other technical advice that we've recommended. So make sure this is done and you're aware of how to do that. Okay, so please ensure that this is not the first time you've seen it on Monday morning at 7:30 or 7:45. Okay, please make sure you're aware of all this stuff ahead of time. So that's what's going to happen if certain things pop up on the day in the exam. Please make sure call me, email me. If not, go into IT and we'll try to rectify it. Please repeat your office number. I'll just put it in here. 28759. So that's my office number. It will come through to me on the morning if you have any issues. Okay. Now the other course did have a Blackboard Collaborate session open that you can jump into if something happens, but I just figure if you've got IT issues, you're probably also not going to be able to log into that, so I don't think I will bother with that. So when you come at 7:45, the first thing you need to do is declare this. Okay, so you'll have this pop up. This is just like the cover sheet quiz. You just read through it and submit. Now then, depending on how quick you do that, you'll probably have about 13 minutes to wait around till 8 o'clock. So you've got to wait till 8 o'clock, and then when 8 o'clock comes about, you'll then see this portal pop up, the end of trimester exam. That will come up. If it hasn't come up at 8 a.m. sharp, then just do a refresh, okay? And then you're right to go. As I said, if anything comes up during the exam, like it freezes or doesn't give you access, then just do what I instructed, call, email, and then we'll go from there. Any questions about logistics before we move on to content? Guys, anything? Anything that happened in 283? I don't want to get into, I know there was quite a few issues, teething issues with 283, but besides the, not to do with the content of 283's exam, but is there anything that you experienced with the logistics or the running of that exam that you wanted to ask questions about its relevance into this exam? I'll just give you 30 seconds to type. I haven't heard anything about students using Wi-Fi at the university. There's a number of students that accessed the computer labs, so actually the hardware, so the computers. Okay, so Deanna, I knew this one would come up. All right, so look, the university, as you know, it's a okay. The university needs to ensure that the students, because it's a unvigilated exam, there's certain aspects that the university will take from a precautions point of view to know whether a student is possibly breaching academic integrity. So the university has ability to analyze the way that you approach the exam, or when you're actually in the exam, with the possibility of using other tabs and so forth. So they do have a bit of that, but they've also gone with the identifying questions, which essentially are questions that only you will know. And so, as you saw with 2803, there were 2 questions that popped up that asked you a specific question, or 2 questions that technically only you guys would know. Now, for every exam, we'll have these questions, and there's no way around it. So yes, there will be there'll be 2 questions in 2804 exam. Now, because my course has extra time, so for 2803, you had 100 questions in 100 minutes. I looked into whether the 2 questions should be given an extra time, but I was instructed that the fact that my exam has more time than the typical exams, because you have 100 minutes for 80 questions, they said that's sufficient. Okay, so yes, you'll get identify questions. I'm not sure who said that, but in ours, you'll get 100 minutes for your 80 questions, and then there'll be the identifiers as well. Okay, but the identifiers should be very specific to you, and you should be able to answer them pretty quickly. All right, anything else logistically for the exam? Otherwise, we'll get into content. No, we're ready to go? All right. Okay. So what I'll do for the first point is I will share you a whiteboard. So let's just start with content questions, and then we'll jump into the specific modules. So before I'll give you maybe we can do for the next 5-10 minutes on just any questions that you have on content. And we can go through those, and then we can go through, once we've done that, we can go through specifically the modules. I think we'll leave 5, 6, and 7 because we've covered those already in the Blackboard Collaborate sessions through the semester. So we can come to specific content in 4, 3, and 2. I don't think during the week, I don't think you guys really expressed any issues with module 1. Would anyone like to pop up any questions onto this whiteboard screen now on questions that you may have on any content within the exam? Okay, analgesic drugs and chemotherapeutic agents. Well, analgesics, in what particular specifics? You got your opioids. You just need to review the receptors that opioids bind to, okay? And then you've got the other drugs being the NSAIDs, you've got your paracetamol, okay, and then you've got your local blockers, so your local anesthetics, which is like lignocaine. You would give that to block pain for a period of time to do maybe some kind of therapeutic intervention like suturing, or maybe relocating a bone or something, or a fracture. That's short term. You should be able to tell me that the NSAIDs work through primarily inflammation, bradykinin, and then your opioids, we've kind of covered that already. Opioids bind to their specific receptors, endogenous opioid receptors, which then have an effect. I think in your case, for the exam, the ventral horn, what it does at the ventral horn to mitigate the transmission of the nociceptive pathway is what you need to know. Chemotherapeutics, specifically, you've got your cell cycle phase specific and your non-cell cycle phase specific, and then you've got your hormonal and your immune. Those 4 are the basis, but as we saw by the question on Facebook, you've got potentially 6 subcategories that fit in with those, okay? Do we need to know the mechanism of action of ibuprofen? Yes, of course, 100%. 100%. So anyone want to instruct us? How does ibuprofen, how does aspirin, what's the primary function, mechanism of action of how they work from an inflammatory basis? I would be hoping these would be rolling off your tongue by now. Yes, blocking COX, cyclin oxygenase. Very good. Well done, guys. So you will definitely need to know that. And then you go to celecoxib. It's something different with that one. What's specific about celecoxib compared to ibuprofen and aspirin? Yes, it's COX-2 specific. Now, why would you just want to block COX-2 only and not the others? Yeah, that's right. Because remember, when you broadly block COX as an enzyme, you're essentially going to diminish the effects of prostaglandins everywhere. Okay, it's not going to be specific just to your swollen toe. It's going to happen everywhere. So you need to go back and go, well, what does prostaglandins do for you physiologically? You know, from what does it do from a homeostatic point of view? Okay, and once you know that, then you can understand. Then that's where all your side effects come from. What are some common side effects, guys, of blocking COX? Yes, sorry, dorsal horn, not ventral. My apologies. Not motility. Probably not so much acidity. Yes, mucus. Right. So what's the mucus in your stomach for? Protecting the lining, yep, and neutralizing the acid. Well, when you say neutralizing acid, you just mean within that mucus. So remember, the mucus is kind of sticking to the stomach wall, okay? And so if the acid comes close to the stomach wall lining, it would start to burn holes in it. But if you produce a mucus that has bicarbonate in it, like an antacid-like chemical, therefore it's not going to have its acid effect on the wall of the stomach. Now, if you block that, you can probably guess what happens. Prostaglandin is also very important for regulating GFR, so you can probably guess what would happen then if you're not regulating your GFR very well. Anything else that you can think of, guys, side effects of broad cyclooxygenase blockage? Yeah, so you'd have that effect, which may be impacting the bleeding. Okay, that might be useful for part of the use of aspirin, but if you're giving it for another means, then you might be taking into consideration an increased bleeding effect. Good. Moving to the purple. With developmental disorders, do we need to know the diagnostic testing and clinical manifestations? You should know broadly how, if you're talking about congenital and developmental from an embryological fetal point of view, you should know what diagnostic testing is available to be able to diagnose this, yes. I don't want you to go into specifics of all the different types of developmental disorders. You might just want to have a look at the main ones which are listed in the course site, probably being Down syndrome and fetal alcohol syndrome. But yes, you should be aware of the teratogens that are listed. That's right. But I don't expect you to go into all the subtle differences of, septal defects of the heart and things like that. You don't need to know that level, no. Okay, how do we relieve adverse effect of chemotherapy? You just have to, if you're referring to things like the blood cells, you just have to keep an eye on them and then maybe pull back the strength of the chemotherapy. So you might have to miss a couple of weeks of it. You might have to modify the dose. You might have to, just play it. This is the oncologist would be doing this, but just play around with maybe the type of chemotherapy, the dose of the chemotherapy, the duration of the chemotherapy. Maybe miss a couple of weeks to particularly look at the ones that are most detrimental and most serious in complications. So this would be things like white blood cells, red blood cells, platelets. They're the main ones, but you probably, depending on the specifics of, some people get quite serious skin irritation, so they're bleeding, or they get lots of bad sores, so that might have to be pulled back on. Some people get really bad neuropathies, so they get a lot of pain in their fingers. Not so much pain, but sensitivities in their fingers, so they might have to get pulled back. So it really depends on the chemotherapy, but probably modifying the dose, etc., would be how you relieve it. Much else you can do besides that, because I mean, you are essentially given a drug that kills cells. Okay, so it's not something you take. You know, it's there to kill cancer, but it's going to kill your own cells. So it's a balancing act. And I think I said yes. Sorry, the dorsal horn. All right. Any other questions, guys? Okay. What I'll do, I think what I'll do is just give you a bit more information about the learning outcomes of the first 4 modules, because these are pretty much the revision that we need to go through. So just bear with me for 1 second while I load it. Exam, practice exam, study guide. All right, let's start with patho. Okay. All right, so let's just, all we're going to focus on now, let me see if this actually zooms in. Does that zoom in for you guys? Or is it just me? So is it currently zooming in? No, okay. Bummer. All right, but you can see the study guide on your screen. Okay. All right, well, that's unfortunate. We can zoom. Yeah, so if everyone can kind of zoom in a bit just so you can see the writing a bit clearer. So, yeah, that's right. Maybe it's my old age. Not sure why this is not selecting it. Oh, okay. Let's go to module 1. Okay, so module 1, cell path. Okay, there's 2 things you need to know for the pathophysiology aspect of your exam. Okay, so just to reassure you, because I know this came up on the Facebook yesterday, the study guide, the students suggested the study guide for 283 didn't match the exam. I'm telling you now, I literally went through the exam with this next to me and I ensured that it married up. Another thing that I can tell you is Ewan and I went through the exam this morning and we question by question to ensure that A, the questions weren't ambiguous and they were completely relevant to these learning outcomes that in your study guide, and also essentially, well, that's the main one, just to make sure that the answers were definitively only one correct. Another thing that I will just tell you is that for your exam, there are a stratification in difficulty. So the vast majority, so top of my head, I'd say something like 60 to 70% of the exam is pretty much questions of a pass kind of level of difficulty. There's a couple of HD questions thrown in there, but it's only a couple, and then there's a few more credit kind of distinction levels. So there will be a range of difficulties. The vast majority we've put in the green section, which just suggests that these are pretty simple things that should be known, whereas there's always going to be a couple that are, at the high HD level. Okay, so just to let you know, that's how we've set it out. It's been extensively moderated by all the science people. So that's Grant, Mike, myself, Ewan. So we've got all the science people making sure that the content is correct, and then we've sent it on to all the clinical people, both medical doctors and nurses, to ensure that the clinical relevancy is correct. Okay, so ensure this is just to give you that reassurance that it's been heavily moderated. I can only speak to my exam. I can't talk to you about the other exams. All right, so coming on to module 1. So what you can see here is you need to be able to explain. This is what this module is asking. Sorry, this is what this learning outcome is asking you to do. Explain it. So you need to be able to explain this process. Okay. So don't necessarily think that you're just going to be asked a definition of what inflammation is. All right. So if you're expecting a question to pop up and say, please define, or which of these 4 definitions of inflammation is correct, you might be rudely shocked, because that is not being able to explain something. That's to define something, okay? Or even describe something. Once you explain, it's an expectation that you understand it, okay? So what it's really expecting you to know is both the cellular and the vascular phases of inflammation, okay? So you need to know what's happening to cause, at a blood vessel level, to lead to the 5 cardinal signs. You need to be able to explain it. And same at the cellular. What are the cells doing in that region of inflammation, okay, to bring in the cardinal signs? So what are the mast cells doing? What are the macrophages doing? What are the neutrophils doing? What's the blood vessels doing? What's causing the blood vessel to dilate? What's causing the blood vessel to become leaky? Okay, these are the things you need to be knowing. Okay, any questions with that? Just pop them in as we go. Okay, and then again, you've got to go compare and contrast. So this is again expecting you to understand depth, to know the difference between acute and chronic. Okay, but not only in the context of knowing the difference in acute or chronic, but you need to know what the causes of their difference are, what are their mechanistic differences are, what are their cells involved difference and similarities are. Okay, and then finally, what's the outcome difference between acute and chronic? Okay, so there's a number of things you need to know to be able to complete this learning outcome, and the questions will be assessing this. Again, it won't be just saying, please define chronic inflammation. It will ask you something specific, and it might then suggest, what cell would you expect to see if this is now a chronic type of infection? If you were to do not a biopsy, but let's just say you were to do a microscopic analysis of the wound, a chronic inflamed wound, what cells would you might expect to see at the tissue level to confirm it's chronic, not acute? Okay, hopefully what you can see from that example, that you need to understand certain things to be able to answer the question. Okay, moving on. Microbiology, so describe the developmental stages. So you need to know, in terms of an infection, you have this course of infection. Now, who could tell me the 4 main stages that happen in a typical infection? So this could be a viral, could be a bacterial, it could be a fungal, it could be a protozoa. Yep, so incubation is definitely the first, because the microorganism has to come in and invade you. Then it needs to come up to a sizable amount, okay, and then it needs to bring on the symptoms, okay? So you might get questioned on, with the context, okay? You might get a question in the context of a infection that you know nothing about, but that's irrelevant, okay? So let's just say it asks you a question about TB, okay? We don't expect you to know tuberculosis, but it might ask you in terms of, here are the symptoms of tuberculosis, okay? What developmental stage is the course of infection in? And you just, all you need to know is, would you expect to see anything in the incubation phase? What kind of things would you expect to see in the prodromal phase? What things may you expect to see in the acute phase? And then what would you expect to see in the resolution or kind of sepsis phase, okay? It doesn't really matter the specifics of the infection, because the same kind of four stages occur. Okay, you should also know what would justify a local infection versus systemic infection effects, okay, or clinical manifestations. So if a person was to have TB, okay, again, I don't expect you to know of TB, but I'm sure you guys have a bit of an understanding of what TB might be. What part of the body is generally affected with TB? Tuberculosis. The lungs. Okay. So you would probably predict the local effects are the ones that are going to be affecting the lungs, whereas the systemic effects would be because the infection is in the lungs, right? But if you start to get systemic effects, like, you know, blood pressure's different, heart rate's different, respiratory rate's different, or they get a fever, okay? These are systemic effects, or fatigue, or myalgia, okay? This is not local to the infection, it's actually elsewhere in the body. Okay, now we put some more focus on just the bacteria, okay? Because bacteria generally are the ones you're going to encounter most frequently as a nurse. So you need to know the differences, okay, between a bacterial cell and a human cell. And I'll tell you now, the main differences form within 4 main categories. And this is nice because this is where the antibiotics work, okay? Now, you do need to know the broad differences between a gram-positive and gram-negative bacteria. So what structurally, morphologically, things do they have that will discern them, make difference between them, okay? And this might bring in importance to your antibiotics or the possibility of exo versus endotoxic things that can be produced by the different types. Okay, now we go into immunology. If you've got questions on the microbiology, stop me at any point, okay? Moving on to immunology. All right, so there was a couple of you that did mention that you wanted to know the difference between innate and adaptive. So this would be only really this learning outcome here. So basically the point of the immune system is to prevent you getting infected. So it's to prevent microbiology. It's happening in the first learning outcome in module 2. So it's to prevent that happening. Okay, now your immune system has a number of things in its arsenal to prevent you getting infected by microorganisms. It's got things that are always there and will do the same thing as a reaction regardless of what the microorganism is, okay? And this is called the innate immune system. So this can be broken into 3 kind of parts. It could be anatomy, so you've got certain anatomical structures that prevent you being bombarded and breached, your body being breached by microorganisms. Can a few of you just pop in some anatomy or anatomical structures that your body has in place to stop, let's say, bacteria? Yep, skin. So just for a bit of extra marks, what is it about skin that makes it so good to prevent bacteria getting in? You could probably give me 2 things that the skin has. Keratin, so it's got a really tough protein. Okay. What else does skin have to stop? Yeah, oil's good. Sweat, yep. So it's pH, very good. So there's a number of things on the outside of your skin that stops bacteria either growing too excessively or getting through. Okay. And then you've got mucus. Where would you find mucus? So you don't so much have mucus on your skin, but where do you have mucus? And don't say mucous membranes. Yeah, throat, nose, good. Yep, good. Yep, GIT, well done. So the mucus also helps prevent that happening, okay? Then you got eyes, you've got your tears, nose, you've got your mucus and snot and hairs, ears, you've got wax, okay? Anywhere where you think there's vulnerability for things to come in and invade, you've got certain anatomical barriers, okay? Now you go to physiological barriers, still innate, very important you to know. This is still innate, okay? It's still going to be there regardless of whether it's viral, whether it's bacterial, whether it's fungal. What physiological, so what processes do you have that can kick in? Defecation's probably a bit more of just getting rid of the food that you don't really have. Inflammation's good. Okay, fever's also good. Okay, there's some good examples. And now we go to the cells. Okay, so what cells do you have in your innate immune system that will do the same regardless? Yes, neutrophils. What are neutrophils essentially? Neutrophils are, they are phagocytes, which means what to you? They, not so much bacteria eating, they just eat things. Eat anything they feel that is harmful. It could be a bacteria, it could be an infected cell, it could be a cancer cell, it could be a bit of foreign stuff. Okay, it will just, they'll just come along and eat it. Okay, but they don't specify, they don't specifically go, no, I'm just here to eat viruses, thanks. I don't want to eat bacteria, they're gross. Okay, they just eat anything. Okay, so you've got these white blood cells that will kind of do the same regardless. Okay, you've got natural killer cells. They're also non-specific. They're innate. They're just going to go around, and by their name would suggest that they just kill things. Okay, so that's your innate system. So hopefully that makes sense. Okay, now you go to the adaptive, which means now you've got to take a bit more time to be specific to what you're trying to kill off. Okay, so now we bring in a different white blood cell. Okay, what are the white blood cells? And this is very important. You need to put this into your brain now. What are the white blood cells that are just for adaptive? They're only lymphocytes. Okay, they're only lymphocytes. That's it. None of the others. Okay? Okay, so now let's just chuck across to a whiteboard. So, nope. Can you see the whiteboard? Okay. All right. I'm not sure why the pencil's not been highlighted anyway. So let's say, okay, this is going to be hard, but I'll just try anyway. Okay, here's I shouldn't say COVID. What's the virus? This will test you. What's the virus that causes COVID-19? I want to be impressed. COVID-2. SARS-CoV-2. Yeah. All right. So here's your virus particle coming in. Can everyone see that red kind of flat cloud with a blue bit of RNA in it? Okay. So, and here's a cell. Let's say it's going to, yeah, I told you I can't draw. Okay. So let's say this virus now infects this particular cell. Okay. So essentially what it's going to do is it's going to come in and invade and take over it. Okay. Now, this is very simplified, but essentially it will just take over the cell and trick the cell to then copy heaps and heaps and heaps of viruses. Okay, I'm only going to focus on one cell here. Now, if that cell, when it becomes infected, there is a possibility that that cell can give an indication to your immune system that says, hey, there's some serious problem with me. I really probably should get killed off, please. So what would happen is a phagocyte, to simplify it, a phagocyte will come along. Okay. This is a phagocyte. And this is recorded, yes, unfortunately. And eat it. Okay? So it would come up, engulf it, put it inside its mouth like so, and then put it in like that. Okay? Following so far? Now, it will, just like we do, it will digest that cell and the virus inside it. Okay, so I'm going to get rid of that. And then the cell, what the cell will do is it will chomp it all up. Okay, and then what it will do is has some receptors on it. Some receptors on it, which they call histocompatibility receptors. And what it will do is, I probably shouldn't have made that receptor red because the virus was red, but anyway, I'll make the virus blue. So what it will do is it will chunk up that virus, chop it up into little spots, and put a bit of that virus on the outside of this cell. Okay, so this will then make that cell what we call an antigen presenting cell. So macrophages can do this, not neutrophils can do this, dendritic cells can do this. So they can chop something up and then they can present the antigen. Okay, which means now the antigen, in this case, it could be a little spike of that COVID-2, okay, which recognizes almost like, let's just think of it like a flag, like this. It's like the nation flag of that virus, okay? And then what happens? Now it becomes complicated, but I'll just try and simplify as best I can. A cell comes along, which we call a T cell, and particularly a T helper cell, will bind, okay, to here, to the histocompatibility receptor and say, oh, there's an interesting antigen you've got here, which then makes that particular type of T cell selected for, and then what it will do is it will clone itself and just become specific to that antigen, okay? So it will do that, so it will just keep copying. So this is going to take days and days and days to happen, all right? Now, at the same time, it will come into contact with a B cell, okay? So particularly a naive B cell, okay? And it will also communicate with the B cell and say, hey, B cell, I've just come into contact with this really crazy antigen that I've never seen before, but I think it's coming from a crazy new virus. So can you please help me here? And the B cell, only the right B cell that has within its arsenal a antibody, okay, that fits, okay, perfectly for that antigen, okay? And that's where it would fit, like there, right? So the two of these guys communicate and they chat together, and they then decide that this B cell is most appropriate for that antigen, okay? So now this B cell, before this it was what we call naive, okay? But now it is selected for, so now it's been activated. So now it's going to differentiate into two kind of categories. One being a plasma cell, okay, and the other being a memory cell, okay? Are we all following? So the plasma cell, what that will do is it will differentiate and it will just become an antibody factory. So all it will do now for hours and days is just pump out antibodies and antibodies and antibodies and antibodies specific to what, people? What is this antibody that it's been producing at high quantities specific to? There's only one thing, let's just say simplistically, that can sit at the end of the antibody. What is the antigen that it will only fit to? Anyone? Everyone's fallen asleep. The antibody that this plasma cell has now produced that's specific to is this antigen here, which is the antigen to the virus COVID-2, okay? So now, anytime there's a viral particle, okay, so let's say here's the virus being SARS-CoV-2, there's the antigen or the spike that's on the bacteria, on the virus. So that's telling you that these antibodies that you're just pumping out from the plasma cells will be floating along in the blood, and then once they bind to the receptor on the outside of the virus, what will happen is a siren will go off. Not really, but you get the point. will start to flash like that. And what will come along are the things that will do the killing. So this could be a T cell that's cytotoxic, which then will kill it. Okay, or you might have a natural killer cell that will come along and kill it. Okay, or you have the phagocytes to come along and start killing it. And this gives the immune reaction that's only specific, hence why it's called adaptive, only specific in this case for the SARS virus. And that's why it's very adaptive, and it's going to take a long time. It doesn't happen in hours or even days. It's going to take days to weeks, and this is why people that are infected, it may take them 10 to 12 days to knock it off, depending on how serious they get it, okay? But with the crux of, and this is what we're trying to develop now, is the vaccine, because ultimately you want to have memory, okay? So it's the B cells that produce memory cells, which means once you get rid of the virus and you've killed it off, and then you've gone, let's say, a year later, and you don't have many more antibodies or plasma cells anymore, you still have a few memory cells around, which are just floating around the body. So just in case the virus comes back, you're already primed, ready to go, and you can deal with it much quicker. Okay, so that's hopefully illustrating to you the adaptive immune system. It's very complicated, but hopefully you get a better understanding now of what it kind of means. And this is the system. So antibodies are the big part of the adaptive immune system, because the antibodies are specific to the antigen, and the antigen is specific to the infection. Okay, whereas an innate system is not. It's just going to react the same regardless. So when a person first gets infected with COVID, the first reaction they get is inflammation, because it's non-specific. They get fever, it's non-specific. Okay, they get lung consolidation, it's non-specific. But it's only when you bring in your antibodies and your other lymphocytes is when you can specifically start to kill it off. Okay, hopefully that makes sense. So let's go back to the study guide now. So that is your immunology. Now, when you're focused on these two, okay, so this is a specific type of immune reaction now. So we've got the hyposensitive immune responses. So what you need to know are the four: type 1, type 2, type 3, type 4. In each type, you should know what are the main cells involved, what are the main antibodies involved, okay, and an example of a disease condition that causes it. So type 1, classic, is the hay fever or anaphylaxis. Okay, 2, classic would be given the wrong blood. Okay, wrong transfusion. Type 3, a good example is this is an immune antibody complex, and then you get it lodged in your lung or kidney. And type 4, this is more the typical autoimmune disease. Classic example being type 1 diabetes. Okay, so you need to know those 4 types. Then go into anaphylaxis. That's specific to type 1, but I want you to focus a bit more heavily on this one. So you need to know what's the pathophysiology of anaphylaxis. So what's actually happening to cause the clinical manifestations. And who can tell me maybe 3 common clinical manifestations of anaphylaxis? So if you had a person with anaphylaxis, yes, itchy, hypotension, what's swelling, Maddie? You've got to be specific there. Rash, yeah, that would go with the bronchoconstriction, yeah. So your airways are swelling, okay? So airways start to swell up. If you have your airways swelling up, you're gonna, as Nina said, you're gonna have dyspnea, difficulty breathing. And as Leah said, you're gonna have wheezing, okay? Yeah, you might feel something's about to happen, but it's probably not the best symptom to go off. Okay, so, and just to test you guys, what is the cell What's the cell that's the biggest player with anaphylaxis? Mast cell, brilliant. And what's the, with mast cell, what's the chemical release? Brilliant. So you guys know it. I don't know why I'm here. You should be teaching each other. All right, module 4. Okay, now you got to know carcinogenesis. So you need to know the stages of carcinogenesis. Okay. Now, who can tell me what's the crux of a neoplasm? What's happening at the cellular level to cause a tumor, to cause a growth? What are the cells? What's the cell group essentially have to have as a characteristic? There's 2 aspects to it. Yep, and you both got it. And it's got one, Lee's got the other. Multiplying, so they're copying each other ridiculously fast and a lot. And as Annette said, they live forever, which means they're immortal. Okay, which basically tells you they just keep copying and copying. Remember, cancer just comes from one rogue cell. Okay, so it has daughters and then daughters and then daughters and daughters and daughters and daughters. Now, eventually, if this was a normal cell, it eventually would get so old that you can't have any more daughters and everything will stop. But with cancer, it has the ability just to be immortalized. It just keeps having daughters for thousands of generations, probably more, hundreds of thousands, maybe millions. Okay? So the way it can do this is it can turn its cell cycle off. Okay, so it turns the genes on that turns the cell cycle on. It turns the genes off that would normally turn off the cell cycle. Okay, it would turn the genes off that normally would kill a cell that thinks it's got some problems with it. It would turn the genes on that would allow the cell to leave the area. Okay, it would turn the genes on that would produce an enzyme called telomerase, which allows it to live forever. So there's a number of mutations that have to happen at the cell to then lead it to being immortalized. That means live forever and copy itself rapidly. Okay. Now that's carcinogenesis, but then what increases the risk of this happening? What do you reckon? Anyone? Yep, radiation's good. So this is these carcinogenic things that would increase the likelihood of these mutations occurring. So radiation's good, smoking's good. Yep, some hormones good. Chemicals, you just need to be a bit more specific because everything's chemical. Water's a chemical. Okay, it's just hydrogen and nitrogen, but it's chemical. You just got to be a bit more specific, yeah. Toxin, again, be specific. I don't expect too much, but just be a bit. Luckily, it's not any short answers here. Viruses, yep, good. Okay, and then we go into the, where are we? Diagnostics. I think we've done this. I think you know the different diagnostic assessments and the different ways to evaluate cancer prognosis and whether it's spread. I think you know this. Okay, and I think you know the clinical manifestations of typical cancer. Again, obviously it would depend on where the cancer is. Okay, so what would be, let's just say, a clinical sign of bowel cancer? Does anyone want to pop one in? What about for breast cancer? Blood in stool, good. What about breast cancer? Now, as I've said before, yep, lump. What about lung cancer? Like I've said, I don't expect you to know the specifics, but pretty much you can make sense by knowing the area. You can pretty much guess to coughing up blood. What about brain cancer? Yeah, headaches, seizures. Well done. Excellent work. All right, let's move on to the last one now. So I'll get rid of that, and I'll go here, and we'll put out the study guide for pharmacology. Okay, just bear with me while it's loading. All right, so again, now, so this is why I think there's a question earlier on, do we need to know NSAIDs? This is the only thing you need to know from the module 1 in pharmacology, is how the drugs work for inflammation. So you must know how the NSAIDs work, you must know how the steroids work to get rid of or to mitigate inflammation. It's a must. So in terms of the drugs, you need to know your aspirin, your ibuprofen, your celecoxib, and then the steroids, you need to know your corticosteroids. Not only do you need to know how they work, okay, you've already told me, I mean, the majority of them are working with COX enzymes, but you also need to know when they're predominantly used and their common side effects, which we've kind of already went through. So that's pretty straightforward, I think, okay? You're going to get 5 questions from that, guaranteed, telling you that now, okay? So it's in your best interest to understand it. Moving on, then you should know your antibiotics. Again, you'll get questions on antibiotics. Let's just say amoxicillin, cephalosporin, aminoglycosides. You need to know if a drug like that pops up, you need to know how it works, okay? And then what could be potentially a side effect that would go with it. I don't expect you to know 10, 20, 30, 40 side effects. I just want you to know the main ones that go with antibiotics generically, because there's quite a lot. There's a lot of the antibiotics have the same side effects, but you should just know the few exceptions that have really nasty side effects that you need to be mindful of, like the renotoxicity or the ototoxicity, okay, or the ones that could cause anaphylaxis. You know, just the ones that the side effects that are listed in the course site would be sufficient. And you should also know how resistance comes about. Yeah, I'm not going to ask you about the hairy tongue one. It's a bit specific, but it's good that you know that, Amanda. All right, module 3. Okay, so you need to know when it comes to the immunosuppressive agents, okay, listed in the course site. You just need to know how they work, okay? And then you need to know the side effects. So with corticosteroids, you'll get asked questions on corticosteroids and how it works from an anti-inflammatory point of view, but you also need to know how it works from an immunosuppressant view, okay? How does corticosteroids work to suppress your immune system, okay? You should know the main mechanisms listed there, okay? Also, you should understand what would be the reasons for why you want to give or immunosuppressive agents, okay? Now you need to know briefly the different kind of categories of vaccines. I don't want you to go into the nitty-gritty specifics of, you know, the ones that have got 3 things in them, but I just expect you to know there's ones that are attenuated, there's ones with bits of virus in them, there's some that utilize antitoxin ability, okay? And then you need to know the 4 types of immunity. So this is your passive versus active, natural versus synthetic or artificial, okay? And you should know examples that fit into it. So you should know, where does vaccines fit into it? Where does breastfeeding fit into it? Where does giving antibodies fit into it? Where does getting sick yourself fit into it? Okay? And then finally, we go to module 4, and here's you need to know the chemotherapies. So as I said, the main ones you need to know in terms of the category are the ones that are specific to the cell cycle, ones that are non-cell cycle specific, ones that are specific to hormones, and ones are specific to immune modulating cancer treatments and the side effects that go with it. Now, I don't expect you to know the specifics for those 4 different categories. I just want you to tell me the side effects that go with chemotherapy very generally. So can someone tell me 4 common side effects of hair loss? Definitely. Nausea, fatigue, So you don't feel like eating? Yes, you're missing out important ones. Yeah, I'm not sure cachexia is a hard one. Cachexia could actually come from the cancer necessarily than the drug. I mean, you might get cachexia if you're not eating. Yeah, early menopause, yes. So remember, you just got to think. Always go back to first principles. Chemotherapeutics are targeting highly mitotic cells. So you just got to think, what cells in the body normally have high mitotic turnover? So what cell type is potentially the most mitotically active cell in your body? What cell do you make 2 million per second? That's a lot. Think about that. 2 million cells a second. Give me a cell type. Red blood cells, right. So, and where are red blood cells made? Where do red blood cells come from? Bone marrow, very good. So a stem cell in your bone marrow. What cell type? 4 million cells per second. So dark red blood cells. Anyone? No, platelets. Platelets. What do platelets do, people? Right, very good. And then maybe bring it back a bit, 100,000 cells a second is your white blood cells. They're all coming from the stem cell. So if you stop the stem cells in your bone copying as frequently, what are your 3 side effects going to be just based on those cells? Anemia, infection, bleeding. Brilliant. It would be the opposite. You wouldn't get thrombosis, you'd get bleeding. You wouldn't get clotting, you'd be the opposite. Brilliant, guys. Well done. So that's essentially one of the most important things you'll be regulating as a nurse in oncology. Their infections, their anemia, their bleeding risk. Sure, it's important to know that their hair's fallen out, but it's probably not the end of the world, okay? It's definitely not as important as the 3 you just mentioned, okay? Nausea and vomiting, yep, horrible, but it can be managed. Those other 3, life-threatening if you get it wrong, okay? And then diagnostics. What ways, so this kind of also fits back into the patho section. You know, how would you diagnose cancer? Okay, how would you evaluate it? And what are the generic treatments? Okay, well, you should know there's probably, you should probably tell me 5 different ways cancers can be diagnosed. 5 different diagnostic approaches you can use to diagnose. Again, lab test too general, doesn't mean anything. Yes, imaging, very good. Pet, you ask your pet, Lee. So, you have your pet dog. Actually, you know what? I've heard that dogs can potentially know that people have cancer, so you maybe not too wrongly. PET scans, yes. So that positive something transmission, electron transmission. Okay, good. Brilliant. Red cells, probably not the most accurate. It could be in your, toolbox, but the big ones would be, you got your markers, you got your scans, okay, you got physical examinations, but by far the most specific, most sensitive form, I should say specific, most specific form of diagnostic for any cancer is biopsy. Brilliant. It doesn't matter how you get that biopsy, okay, it's always biopsy. So whether it's excision, whether it's needle, whether it's, I'm trying to think of other ways you can get the biopsy. Well, I guess you can get it in a stool for bowel cancer. It's probably not the best, but that's kind of how they do the screening there. Okay, brilliant, guys. That's really it. Any final questions? So I want to reassure you, if you know the study guide and you can do what's on there, you'll do well in the exam. Okay? I do not want to be tricking you. Okay? I, as a convener, want you all to pass. I want you to get all HDs. So it's against my best interest to make the exam hard and you fail. Really, it is. But I have to, at the same time, ensure that you're competent to go into third year. I don't want the third year conveners going and getting angry at me and saying, Matt, what are these students that don't even know the difference between a corticosteroid and an NSAID? All right, thanks, guys. If anyone does not make 50% overall after this exam, well, you basically need 50% out of your 3 assessment items to pass the course. That's ballpark. Okay, so ballpark speaking, if you get 50% out of the 3 assessment items, it's very, 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 very likely you'll pass the course. I've never seen it below 50%. Okay, so historically, if you get about a 50%, you'll pass the course based on those 3 assessment items, which is the quizzes, the concept map, and the final exam. All right, I will be periodically checking the Facebook over the weekend. Probably not so much email, but if you've got questions, just pop them up on Facebook, and I'll do what I can. Good luck. Please. Please make sure you're on the course site at 7:30. Don't be logging in just before 8 o'clock, okay? Promise me that, okay? And you know what to do if anything occurs, okay? So first port of call, phone the office. Second, email me. Third, IT. But I don't envision any problems. Make sure you read all those things that we've instructed you to read before the exam, just so you're all prepared, ready to go. Otherwise, have a hopefully non-stressful weekend, and I will see you bright and early Monday morning. Be good and keep smiling.